Hi, and welcome back. Today I'm going to be discussing Chapter 22, The Neurobiology of Mental Illness. In today's chapter, we're going to talk about approaches and challenges to studying and treating mental illness. We'll talk about some of the history and theories behind this practice, anxiety disorders, affective disorders, schizophrenia, some disorders that aren't referenced in your book, as well as treatments and neural correlates. So, how do scientists and psychiatrists diagnose mental illness or mental disorders? The DSM-5, known as the Diagnostic and Statistical Manual of Mental Disorders, published by the American Psychiatric Association, helps psychiatrists and clinicians to diagnose psychiatric disorders. So, what constitutes a mental illness or a disorder? Well, this is a clinically significant disturbance in cognition or emotion that can lead to distress or disability. Ultimately, it affects a person's quality of life. And elements of good diagnosis include a cause, a treatment, and a prognosis. There are several different branches of neuroscience and psychology that you could pursue in a future career. So I'm just going to list a few examples here. First, I'm going to talk about neurology. This is the diagnosis and treatment of nervous system disorders. Now, neurologists need to go to medical school to practice neurology, and they focus on the physiological processes that impact the brain or nervous system. And a great example of a disease that they would study and treat would be aphasia. Additionally, psychiatry is the diagnosis and treatment of disorders that impact the mind. So a psychiatrist would work with patients with psychiatric disorders, and they particularly focus on aspects of brain function that are disturbed by mental illness. Psychiatrists also can prescribe medications to treat different psychiatric disorders, which is different from a licensed clinician or marriage and family therapist. An additional branch of psychology called neuropsychology is concerned with how cognition and behavior are impacted by the brain and nervous system. So if you're interested in studying any of this in the future, or maybe considering one of these branches of neuroscience and psychology in your future career, you can always ask me, and I'm happy to help you walk through this. There have been several approaches to studying mental illnesses, and many psychologists and psychiatrists have worked hard to try to create different theories regarding it, such as Sigmund Freud. His theory of psychoanalysis focused on unconscious mental processes and childhood experiences that could potentially shape behavior. He believed that mental illness was a result of conflicting conscious and unconscious desires. Sigmund Freud also proposed that treatment for mental illness involved revealing these unconscious desires. Following the psychoanalysis theory, other psychologists like B.F. Skinner also proposed different mechanisms for which mental illness arose, such as behaviorism. He believed that behaviors are learned responses from the environment, and mental disorders were a result of maladaptive learned behaviors. Ultimately, Skinner's treatment included behavior modification to unlearn maladaptive behaviors. Since Freud and Skinner, of course, we have developed more theories and better models to study mental illness. The best way to diagnose and treat mental illnesses is with the biopsychosocial model. This model is a really great way for us to identify and diagnose psychiatric disorders and find better treatment strategies. This model looks at several different variables that could increase susceptibility to mental illness, such as a person's biology, such as genetic predisposition, neurochemistry or sex, their psychology, such as behavior, personality, their attitudes and beliefs, and also social aspects like education, social support, family backgrounds, and socioeconomic status. Ultimately, there are several different factors that could potentially increase a person's susceptibility to psychiatric disorders, and thus, this biopsychosocial model is interdisciplinary, and this is the best way to diagnose and treat mental illness. Modern technology has now given scientists the opportunity to study a path from genes to treatments for psychiatric disorders. Gene discovery is a key tool in understanding how specific gene mutations or single nucleotide polymorphisms could potentially increase a person's risk of disease. Searching the DNA of individuals with a psychiatric disease may reveal causative gene mutations that can be reproduced in genetically engineered mice. Mouse disease models are an incredible way for us to determine how the brain functions differently in association with these mutations. And so, thus, there are several different mouse models that can be utilized to model really any disease, which is pretty incredible. And disease pathophysiology can be studied in these animals. In these animal models, we can create targeted drug therapy and study how different drugs can be utilized to potentially mitigate symptoms and the biochemical processes underlying specific psychiatric disorders. Now, drug development obviously takes years, and usually drug development begins in cell lines and in mouse disease models, and then after years of experimentation, it can be utilized in human clinical trials to develop new medications to treat psychiatric disorders or neurological diseases. There are several different psychiatric disorders, specifically anxiety disorders, that are discussed in the textbook, such as panic disorder, generalized phobias, specific phobias like social phobia or agoraphobia, and generalized anxiety disorder. And we're particularly going to focus on generalized anxiety disorder and panic disorder. Approximately 25% of the human population will have an anxiety disorder in their lifetime. This is pretty high prevalence. It's mostly prevalent in females, with about 30% of females experiencing anxiety disorders compared to males. That's approximately about 19.2%. And anxiety is primarily future-oriented, resulting in a negative mood state. Approximately 3.1% of the population is diagnosed with generalized anxiety disorder. This is the most commonly diagnosed disorder, which I'll refer to as GAD, and it's more prevalent in women than men. GAD brings on symptoms like uncontrollable worry, nervousness, insomnia, inability to concentrate, irritability, and indecisiveness. These symptoms typically need to last for about 6 months before a person is diagnosed with GAD. Now, GAD affects several different areas of the brain. And so we're primarily going to focus on the prefrontal cortex, the anterior cingulate cortex shown here in green, and the amygdala shown here in red. Now, all three of these brain areas are largely affected by generalized anxiety disorder. And previous research has found that there's decreased connectivity between the prefrontal cortex and the anterior cingulate cortex, as well as the amygdala. And in previous lectures, we've talked a lot about how the amygdala plays an important role in processing and storing our emotional memories. Activation of the amygdala has also been shown to trigger the fight or flight response and the HPA axis. So ultimately, generalized anxiety disorder can affect these key brain regions. And it's often comorbid with depression, which we're going to focus on major depressive disorder in a little bit. Previous studies in human subjects has demonstrated that chronic stress and anxiety is associated with structural degeneration and impaired functioning of both the hippocampus and the prefrontal cortex. So thus, this could account for increased risk of developing several different neuropsychiatric disorders, such as depression, or even other neurological or neurodegenerative diseases like dementia. So if you want to learn more about the connection between chronic anxiety and stress in these brain areas, you can take a look at this paper that I've posted right here. As I mentioned previously, different gene mutations or single nucleotide polymorphisms could potentially increase a person's risk of a different disease or psychiatric disorder. And that's the case with this example I'm going to share. It's called the BDNF Val66 Met polymorphism. I know that's a mouthful, but BDNF, if you remember, stands for brain-derived neurotrophic factor. Now, this gene is very important, and when this gene is expressed, it can help increase learning and memory consolidation. So ultimately, if you remember, I've talked about how BDNF has been shown to increase after exercise or after eating a healthy diet. This single nucleotide polymorphism that I'm going to talk about, called the Val66 Met, has been associated with increased anxiety. And so if a person was homozygous for the 66 Met allele, meaning they carried both alleles, this actually only occurs in about 10% of the population. This homozygous 66 Met allele has been associated with increased anxiety-related facets on the construct harm avoidance, which really primarily looks at anticipatory worry and fear of uncertainty. And interestingly, researchers have found that women are more susceptible to anxiety if they are a carrier for this polymorphism. So if you're interested in investigating this, you can take a look at these two research papers I attached right here. Also, let's quickly talk about panic disorder. This is a type of anxiety disorder that only affects about 2% of the population. It affects more women than men typically. And this brings on terrible feelings of intense terror, heart palpitations, sweating, shortness of breath, chest pain, nausea, dizziness, and trembling. So these are all symptoms of a panic attack. And so interestingly, prior research has found that panic attacks are associated with decreased frontal lobe activity and increased activity in the right amygdala. Also, I want to note that there's decreased prefrontal cortex inhibition of this fear circuit. Panic disorder is typically comorbid with depression and substance abuse disorders, as substance abuse is primarily a coping mechanism for panic disorder. Another psychiatric disorder we'll talk about that is under this umbrella of anxiety disorders is post-traumatic stress disorder, or PTSD. Now, you're probably familiar with this from your other psychology courses, but PTSD affects about 3.5% of the population. And this results from a traumatic experience. This could be a direct experience, witnessing a traumatic experience in a person near you, learning about a relative or close friend who had the experience, or potentially repeated exposure to this experience. This could typically be seen in first responders or law enforcement. Symptoms of PTSD include increased anxiety and intrusive memories and flashbacks of the traumatic experience, irritability, emotional detachment, and increased sensitivity to stimulation. People that have PTSD can become overstimulated very easily. Researchers have found that PTSD affects various brain areas, including the prefrontal and cingulate cortex, the thalamus, the hippocampus, and the amygdala. So many of these areas are in the limbic lobe. Research on PTSD has demonstrated that epinephrine and norepinephrine are potentially released during a traumatic experience. This makes sense because during a traumatic emotional experience, the sympathetic nervous system, our fight or flight response, is typically activated. Based on your prior knowledge from this course, you should understand that neurons are typically releasing epinephrine and norepinephrine during the sympathetic nervous system response. Thus, these two neurotransmitters could potentially boost emotional memory formation via the beta-adrenergic system during a traumatic experience. Thus, researchers have been investigating potential treatments that could target these different catecholamines. One potential treatment is a beta blocker that's called propranolol. Propranolol counters these effects by competing with catecholamines at the beta-adrenergic receptors and potentially reducing emotional memory formation associated with a traumatic experience. Now let's talk more about this on the next slide. In clinical settings, beta blockers like propranolol could be administered to potentially reduce the memories associated with trauma. So, what exactly do I mean by this? Well, essentially, a clinician could ask a patient or client to reactivate a traumatic experience, maybe by sharing the experience and talking about it out loud. Usually, talking about an experience and feels like reliving that experience, right? That can usually bring on physiological responses like increased heart rate or blood pressure. Interestingly, prior research, as shown in this paper by Young and Butcher, published in 2020, in prior research, scientists have been able to demonstrate that beta blockers administered prior to the reactivation of a traumatic experience could actually decrease the severity of PTSD symptoms and the physiological responses. Additionally, beta blockers have been shown to improve cognitive performance in people with PTSD. So ultimately, beta blockers could potentially block the reconsolidation of these very salient traumatic experiences, which could ultimately help people mitigate their symptoms of PTSD. So if you want to take a look at these papers, I've posted the links here and check them out if you're interested in looking more at beta blockers as a potential treatment for PTSD. Under that umbrella of anxiety disorders is another disorder known as obsessive compulsive disorder, which is commonly referred to in the field of psychology as OCD. Now, this only affects about 2% of the population, and there are no known sex differences. Symptoms of OCD include intrusive thoughts and compulsive behaviors. It's often comorbid with anxiety. There are several different brain areas that are involved in obsessive compulsive disorder, like an overactive basal ganglia and caudate nucleus, as shown here in this image. And we know that the caudate nucleus is a part of the basal ganglia, and it also plays a role in impulse control. Also, the prefrontal cortex is largely affected in OCD, which ultimately increases anxiety-like behavior. Thus, other treatments that are typically utilized for disorders like generalized anxiety disorder are also utilized to treat OCD. This could include cognitive behavioral therapy to modify these behaviors, such as compulsive behaviors, and antidepressants like Prozac. Scientists are starting to dig into gene mutations that could potentially be associated with OCD. And so in this image over here, I just want to take a look at this before we talk about this study. You can see that many of the areas that are activated in generalized anxiety disorder or PTSD are also activated from OCD. We've talked about the frontal cortex today. We've also talked about the anterior cingulate cortex. And ultimately, OCD induces overactivation of the basal ganglia and the caudate nucleus. Well, a new study found that there was the presence in some families of a rare mutation in the serotonin transporter gene that was actually associated with a high incidence of OCD. So thus, we now know that serotonin does play a role in the etiology of OCD, although these biological mechanisms are still being further investigated. But ultimately, this rare gene mutation is one way that we can link serotonin deficiency to obsessive compulsive disorder. Now, we've covered the HPA axis a lot in this class. Remember, this is our hypothalamic pituitary adrenal axis, which is really activated during sympathetic nervous system activation during the fight or flight response, known as the stress response. Ultimately, the HPA axis is overactivated in anxiety disorders, which makes sense because if a person is undergoing anxious thoughts or chronic worry, they're likely going to experience stress. Now, we know that the end result of the HPA axis is production of the stress steroid cortisol by the adrenal medulla on the kidneys. and prior studies have investigated how these different hormones could potentially affect anxiety-like behaviors. Now, remember that the hypothalamus has specialized neurosecretory cells in the paraventricular zone, and these neurons, such as parvocellular neurons, release hypophysiotropic hormones, such as corticotropin releasing hormone, or CRH. Now, if you remember, CRH is a chemical messenger that communicates with the anterior pituitary gland. The anterior pituitary gland produces ACTH that goes into the bloodstream and tells the kidneys, specifically the adrenal gland on the kidneys, to produce cortisol in response to stress. Interestingly, prior studies have found that eliminating CRH leads to decreased anxiety-like behaviors in mice. And remember, in my previous lectures, I've talked about how we can measure anxiety-like behavior in different paradigms, such as open field or the elevated zero paradigm. Ultimately, reducing CRH can actually mitigate anxiety-like behaviors. Thus, we know that the HPA axis plays an important role in anxiety disorders. Interestingly, overactivation of the amygdala increases activation of the HPA axis in anxiety disorders, which ultimately results in cortisol release. Sensory information, such as a stressful event or environmental trigger, enters the basolateral nuclei of the amygdala. After it's processed here in the central nucleus, shown here in green, this information essentially activates the amygdala and activates the stress response. This communication between the amygdala and the hypothalamus ultimately activates the HPA axis and the sympathetic nervous system response. During this time, avoidance behaviors can occur, and the diffuse modulatory systems increase vigilance. So both the amygdala and the HPA axis are largely activated during psychiatric disorders. Now that we've talked about all of these symptoms and biological mechanisms underlying different psychiatric disorders, particularly anxiety disorders, let's talk about different treatments. So we've talked about different treatments for anxiety before, but I'll briefly go over them again. We've talked about cognitive behavioral therapy, which focuses on modifying behaviors. We've discussed anxiolytic medications such as benzodiazepines. If you remember, benzodiazepines and barbiturates actually bind to the GABA A receptor, which largely modulates most of the inhibitory responses in postsynaptic neurons across the nervous system. And selective serotonin reuptake inhibitors, SSRIs, are typically prescribed for long-term treatment. As I mentioned previously, benzodiazepines have been utilized for different anxiety disorders, and I do want to note that these are usually short-term treatments because they are very effective for acute anxiety. So they work very rapidly compared to an SSRI. So as you guys remember, we've discussed the GABA A receptor. This is one of the main receptors that regulates inhibition of neurons across the nervous system. And we've talked about how benzodiazepines and barbiturates, as well as ethanol, can bind to the GABA A receptor to increase inhibition of postsynaptic neurons. Benzodiazepines bind to GABA receptors and make GABA more effective in opening its channel and producing inhibition. A typical benzodiazepine, known as Valium, is very highly effective at treating acute anxiety, and this can help to calm down a person very quickly. As I've mentioned previously, different psychiatric disorders can potentially change a person's neurochemistry, and this is very apparent in the context of panic disorder. In this study that we're going to talk about, benzodiazepine sites actually change and fluctuate from panic disorder. So we're going to look at this In this figure from your textbook, we're looking at diminished binding of radioactive benzodiazepine receptors in a patient with panic disorder compared to a healthy subject. So ultimately, in this study, they took a group of participants who were not diagnosed with an anxiety disorder versus participants who were suffering from a panic disorder. The healthy control subject is on the left. The person with panic disorder is shown here on the right. And remember, PET scans allow us to look at brain metabolism and activity. Interestingly, color coding can help us look at benzodiazepine binding sites in the brain. So hotter colors like red indicate that there are more benzodiazepine receptors present, whereas cooler colors indicate that there are fewer. And in this study, especially in the frontal cortex at the top of the scan, we see that there are fewer binding sites for benzodiazepines in the patients with panic disorder compared to healthy control subjects. As we can see, there are more benzodiazepine sites here in a healthy human subjects. So this indicates that in the context of panic disorder, patients may have fewer binding sites for benzodiazepines, which could largely affect this reduced inhibition of postsynaptic neurons that are largely modulated by the GABA A receptor. I know we've already touched a lot on SSRIs in this class, but I just wanted to refresh everyone's memory. So remember that serotonin's actions typically use G protein coupled receptors, and they're ended by reuptake via serotonin transporters. These transporter proteins, as shown right here, play an important role in taking up extra serotonin left in the synaptic cleft and bringing it back to the presynaptic neuron. However, in the context of an anxiety disorder, such as generalized anxiety disorder, we want to increase the amount of serotonin that remains in the synaptic cleft, usually because that person could be struggling with brain-derived serotonin deficiency. SSRIs are a drug that were created to prolong serotonin's actions by blocking the reuptake of serotonin, enhancing its effects at receptors and preventing transporter proteins from taking up that extra serotonin. However, I want to note that these anxiolytic effects of SSRIs are not immediate. It actually takes weeks of regular dosing for an SSRI to work. It literally changes a person's neurochemistry. So ultimately, SSRIs are great for mood disorders, anxiety disorders like OCD or generalized anxiety disorder, but this is a long-term treatment and therapeutic. And so this is different from benzodiazepines that would be utilized as a quick short-term treatment. Interestingly, studies have found other benefits of SSRIs. In studies in rodents, SSRIs have actually been shown to increase glucocorticoid receptors in the brain. And SSRIs may dampen anxiety by enhancing the regulation of CRH producing neurons in the hypothalamus. So if you remember, the neurons that are responsible for increasing CRH, the parvocellular neurosecretory neurons in the paraventricular zone, are largely activating the stress response via the HPA axis. SSRIs can actually enhance the feedback regulation and prevent that overactivation of the HPA axis, thus preventing excess cortisol release. So SSRIs have multiple biological mechanisms that are therapeutic for generalized anxiety disorder. I do want to note that only 50 to 60% of people typically respond to medication and psychotherapy for generalized anxiety disorder treatment. And only a quarter of patients usually experience complete resolution. So thus, in addition to medication and psychotherapy, my research has focused on how nutrition could actually be a key tool in battling anxiety. Plentiful research has demonstrated that nutrition does play a role in anxiety-like behavior in rodents and humans. In diets like the Western diet, which I always refer to as the standard American diet, are typically really high in refined carbohydrates. So this could include white bread, pastries, cupcakes, desserts. And these refined carbohydrates have a high glycemic load. If you remember, the glycemic index is basically a numerical value that can be assigned to a food from 0 to 100, 100 being the highest glycemic index possible. So that would be like pure table sugar. A glycemic load of a food or the glycemic index of a food can help us determine how quickly that food is going to increase or spike your blood glucose levels after eating it. If you have a large, quick spike of blood glucose levels, insulin is going to have to be produced at rapid rates, and it's going to have to work hard to take that newly synthesized glucose and store it in the form of triglycerides or glycogen in your fat and muscles, respectively. So ultimately, this increase in glucose and insulin is associated with an increased risk of obesity. And obese patients actually have a 25% increased risk of developing anxiety or mood disorders like depression. So, as I mentioned before, unhealthy diets like the standard American diet are also associated with reduced serotonin in some regions of the brain. And key takeaways from prior research has demonstrated that high fat, like saturated fat or omega-6, and high carb diets that have a high glycemic load, typically from refined carbs like this cupcake, can change your brain chemistry and potentially increase risk of anxiety. Now that we understand that the Western diet or the standard American diet can potentially increase risk of obesity and anxiety by potentially reducing serotonin, nutrition strategies to increase serotonin could be utilized to relieve anxiety. This is a huge focus for Dr. Uma Naidu, who is a nutritional psychiatrist at Harvard University. If you haven't read her book, This Is Your Brain on Food, I highly recommend it. I utilize this in my nutritional neuroscience course. And Dr. Uma Naidu talks about how multiple studies have looked at tryptophan supplementation and how it could be utilized to increase serotonin. If you remember, tryptophan is an amino acid and precursor for the neurotransmitter serotonin. A high tryptophan diet could potentially raise serotonin levels for the anxious brain. So this has been investigated in both animal and human subjects research. In animals, pure tryptophan can reach the serotonergic system. If you remember, serotonergic neurons live in the brainstem here in the raphe nuclei. In humans, research has found that taking a pure tryptophan supplement can actually increase brain-derived serotonin. So thus, these studies ultimately demonstrate that taking a purified dose of tryptophan can increase serotonin production in the brain. So for those of us who aren't taking purified tryptophan, how else could we increase tryptophan in the body? So interestingly, studies have investigated the effects of tryptophan-rich diets in addition to this purified tryptophan supplementation. And in this study that was discussed by Dr. Uma Naidu in her book, This Is Your Brain on Food, a group of scientists found that a tryptophan-rich diet did reduce anxiety in human subjects. One group of people were assigned to a tryptophan diet that provided 5 milligrams per kilogram of tryptophan, whereas the second group received double the amount. They received 10 milligrams per kilogram of tryptophan. And ultimately, the group that consumed this tryptophan-rich diet did have reduced anxiety. However, I do want to note that studies have shown that purified tryptophan increases brain serotonin levels, whereas tryptophan-rich foods alone do not. Tryptophan-rich foods like chickpeas could only increase peripheral serotonin. So how can you make sure that tryptophan-rich foods get to the brain to increase brain-derived serotonin? Well, ultimately, your eating strategies matter. So let's take a look at this plate for our example. If you were consuming, let's say, turkey with brown rice and broccoli, we know that turkey and other types of poultry are very rich in tryptophan. Well, interestingly, tryptophan is the least abundantly utilized amino acid. The body's going to prioritize the transport of other amino acids to the brain if you were to eat, let's say, turkey by itself. However, if you consume that turkey with broccoli and brown rice, insulin is going to come and take that newly synthesized glucose from carbohydrates in the form of glycogen and send it to the muscles. Those other amino acids also need to be sent to the muscles, which basically prioritizes tryptophan and gives it the opportunity to get up to the brain to help us create brain-derived serotonin. Thus, ultimately, you need to eat tryptophan-rich foods, particularly tryptophan-rich proteins or carbohydrates, with carbs to ensure that they can get up to the brain. A great example that Dr. Uma Naidu gives is chickpeas. Chickpeas she refers to as nature's Prozac, which I love. Prozac is an antidepressant. And chickpeas are very rich in tryptophan, and they're a great source of plant-based protein. But chickpeas should be consumed with a whole grain carbohydrate to ensure that that tryptophan can actually get up to the brain, because those other amino acids will be sent to the muscles, allowing for tryptophan to gain access and transport to the brain. So if you want to learn more about eating strategies like this for psychiatric disorders, I definitely recommend reading this book, which again is part of the curriculum in my class called Nutritional Neuroscience here at Vanguard. Also, I wanted to provide you guys with a list from Dr. Uma Naidu's book. And this basically is a list of foods, vitamins, and minerals that she recommends to combat anxiety. So it's definitely worth investigating these. If you yourself deal with anxiety, or maybe you have a friend or a family member, these are potential ways that they could treat their symptoms of anxiety. But remember to always check with a doctor first to ensure that it's okay to take these different supplements or foods with particular medications. Next, we're going to talk about affective disorders, which are commonly referred to as mood disorders. Here's a list of different affective disorders. We're not going to have time to talk about all of these today, but we are going to talk about major depressive disorder, known as MDD. Seasonal affective disorder was covered in my previous lecture, so I'm not going to talk about that. You can learn more about that in my lecture, chapter 19. We're going to talk about bipolar disorder, and some other disorders you could investigate that we're not going to have time to talk about today include borderline personality disorder and narcissism disorder. Major depressive disorder, known as MDD, is the most common mood disorder. It affects about 6% of the population and is twice as common in women than men. About 20% of the human population will experience a major depressive episode at least once in their life. This could induce symptoms like loss of appetite or potentially increase appetite, lack of sleep or sleeping too much, fatigue, feelings of worthlessness and guilt, a diminished ability to concentrate, and recurrent thoughts of death. So if we take a look here at this graph, this is from the National Institute of Health, and they gathered data in 2021 to look at the prevalence of major depressive episodes in adults in the United States. The y-axis here is showing us the percentage of adults diagnosed with major depressive, the prevalence of adults who had a major depressive episode, and is also divided by sex and age. As you can see, women are at a higher risk of MDD, and the age demographic of 18 to 25 is at the highest risk of major depressive episodes. So ultimately, it is a lot of our younger adults in the United States that are struggling with MDD. MDD is diagnosed after a person's experienced symptoms every day for at least 2 weeks. And sadly, without treatment, MDD usually lasts around 4 to 12 months. Episodes of major depression don't typically last more than 2 years, but unfortunately, the odds of recurrence are very high, about 90%. And previous research has shown that there's hypoactivity, meaning inhibition of neuronal activity in mood regulating areas in the brain. So this could include areas in the limbic lobe or the dopaminergic diffuse modulatory system. So depression involves key brain areas that are very similar to those of generalized anxiety disorder and other anxiety disorders, including the prefrontal cortex shown here, the anterior cingulate cortex shown here in red, and the insula. So in major depressive disorder, we see that there's increased activity in the anterior cingulate cortex. And this decreases with successful treatment such as antidepressants. So in previous chapters, specifically in our Sex in the Brain chapter, we talked about the insula. Remember the insula, as shown right here, plays an important role in lust versus love. So we've only talked about it in that context before. As you can see here, we can look at both the anterior and posterior lobe of the insula in blue and yellow respectively. Essentially, although it's also involved in lust and love, the insula handles sensory, emotional, and decision-making processes, and it can communicate with the thalamus, the brain's key relay station, to bring these sensations into consciousness. There's also evidence that major depression can decrease the connectivity between the thalamus and these other brain areas like the insula. So ultimately, major depression can affect this communication between the thalamus and the insula, and long-term depression has actually been associated with shrinkage or atrophy of the hippocampus. Another affective disorder is bipolar disorder, which I'm sure you're familiar with from your other psychology courses. But today I'm going to talk about 2 of the 3 main types of bipolar disorder, including type 1 and type 2. So bipolar disorder induces episodes of mania and depression. And of course, I've already talked about there are 2 different types, type 1 and type 2. In type 2, people can experience depression in hypomania, which is a less severe form of mania. But ultimately, mania can be described as this distinct period of abnormally elevated or irritable mood. So symptoms of mania include grandiosity, inflated self-esteem, decreased need for sleep, increased talkativeness or flights of ideas, distractibility, and increased goal-directed activity, as well as impaired judgment. Biomechanisms of bipolar disorder first include increased dopamine and norepinephrine release. Increased dopamine and norepinephrine can contribute to mania. Also, researchers found that there's decreased activity in different cortical areas, which we're going to talk about a bit. Interestingly, mania is associated with reduced cortical thickness in some of the frontal lobe areas, which I'm going to talk about on the next slide. Also, another biomechanism of bipolar disorder is increased activity in the amygdala. We know that the amygdala plays a really important role in our emotional memory processing, and ultimately is activated in anxiety disorders and some affective disorders like bipolar disorder. As I mentioned previously, mania is associated with reduced cortical thickness in some of the frontal lobe areas. So in this study by Abe and colleagues, published in 2015, scientists explored the correlations between cortical thickness, so this would include gray matter, the number of nuclei, and the presence of mania in patients with bipolar disorder. And ultimately, the results of this study shown here found that mania was associated with reduced cortical thickness in the frontal lobe. Now let's talk about different treatments for affective disorders like major depressive disorder and bipolar disorder. Ultimately, 2 key diffuse modulatory systems have been targeted to create drugs for affective disorders. And this could include the serotonergic system and the noradrenergic system. So, as I previously mentioned, serotonin and norepinephrine have been linked to affective disorders, and we're going to talk about how these monoamines play a role in the biochemistry of these disorders. So, as I previously mentioned, different monoamines like serotonin and norepinephrine have been implicated in the development of affective disorders like MDD and bipolar disorder. And ultimately, researchers did not understand the biological mechanisms of these psychiatric disorders until the introduction of the monoamine hypothesis. This hypothesis was actually discovered by accident, like many scientific experiments before it, but ultimately, this was discovered while utilizing a drug called Risperidone. And this was initially introduced to control high blood pressure. However, researchers found that about 20% of people who were taking this drug developed severe depression. And ultimately, we now know that Risperidone depletes central catecholamines and serotonin by interfering with their loading into synaptic vesicles. So ultimately, this was one of the identifying pieces of evidence for the monoamine hypothesis. Additionally, in another happy accident, another class of drugs that were initially utilized to treat tuberculosis actually induced increased mood elevation and happiness. Well, these drugs inhibited monoamine oxidase and thus increased mood by blocking that enzymatic degradation of monoamines like serotonin in the synaptic cleft. So ultimately, both of these drugs helped to mobilize this evidence that monoamines like serotonin and norepinephrine are largely affected by different psychiatric disorders. The monoamine hypothesis essentially focused on depression as a consequence of a deficit in one of the diffuse modulatory systems. So we know that depression is implicated in reduced neurotransmitters like serotonin, dopamine, and norepinephrine. And so since we know that mood is closely tied to the levels of monoamine neurotransmitters, we can make drugs to particularly treat these deficits. For example, depression is a consequence of a deficit in one of these key diffuse modulatory systems, and thus drugs have been created to restore those normal levels of neurotransmitters. So now we're going to investigate other treatments for affective disorders. There are several different treatments for affective disorders. Not all of them are pharmacological like you'd expect. I'm going to talk about some treatments that are widely used and some that aren't used a lot. We know that cognitive behavioral therapy, or CBT, is widely used to help modify behaviors, to overcome negative views of oneself. Electroconvulsive therapy has actually been utilized to decrease major depression and other affective disorders. Ultimately, this involves electrical currents that are sent into the brain. The mechanisms of this therapy are not very clear, but we know that it likely affects the hippocampus's regulation of the HPA axis. Finally, the most radical treatment that's not used very often is called deep brain stimulation, or DBS. This involves electrical stimulation of different areas deep in the brain, especially the anterior cingulate cortex. So this is a surgical operation in which these electrodes are actually implanted into the brain and they remain there. They largely stimulate Brodmann's area 25 in the anterior cingulate cortex. And this is done by implanting electrodes that can provide immediate relief for patients with super severe depression. And although this is a promising technique and it has been shown to help people with very major depression, it's a last resort treatment because it's an actual brain surgery, and ongoing studies are going to continue working to look at its effectiveness. I also have a link in the PowerPoint slides for you guys if you want to check out this news story. And the news anchor interviews a few people who have received deep brain stimulation and how it improved their symptoms of major depressive disorder. Other pharmacological treatments for depression include tricyclic compounds. They're named for their chemical structure. So among other actions, they actually block the reuptake of both norepinephrine and serotonin by transporter proteins. SSRIs, we know one that's typically used is sertraline, also known as Zoloft, which act only on serotonin terminals. They're also norepinephrine and serotonin selective reuptake inhibitors. And we've also talked a bit about monoamine oxidase inhibitors in class, but we're going to talk more about their mechanisms on the next slide. Remember, we've talked about monoamine oxidase inhibitors and how they're primarily utilized to prevent enzymatic breakdown of norepinephrine and serotonin. Remember, monoamine oxidase is an enzyme that essentially degrades any excess serotonin and norepinephrine left in the synaptic cleft. And in the case of affective disorders, you would want to block that enzymatic degradation. Also, tricyclics shown up here block their uptake of these important monoamines, and SSRI selectively block serotonin reuptake by blocking those transporter proteins. So there are several different treatments for bipolar disorder, such as antidepressants, but another treatment is also lithium. Now, you've probably heard of lithium as a treatment for bipolar disorder, but you probably don't know how it was discovered. This is an odd story, but an Australian psychiatrist named John Cade discovered lithium's effectiveness for bipolar disorder through serendipitous experiments with guinea pigs in the 1940s. So he was testing urine constituents, but found calming effects of lithium urate in his guinea pigs instead. So further research confirmed that lithium has mood stabilizing effects by affecting neuronal processes. And lithium can affect neurons in several ways. First, it passes through sodium channels freely, and inside neurons, it actually disrupts the turnover of PIP2. And this is crucial for generating second messenger molecules. We know that second messengers can lead to long-term postsynaptic changes. Lithium interferes with adenyl cyclase. Remember that adenyl cyclase is necessary for cyclic AMP production and glycogen synthase kinase. We know that this is super vital for cellular energy and metabolism. So ultimately, these are some of the biological mechanisms in which lithium can help increase mood. Finally, we're going to discuss schizophrenia, which only affects about 1% of the adult population. Essentially, people with schizophrenia have a loss of contact with reality, disruption of thought, mood, and movement, and oscillations between normal and abnormal states. So there are both positive and negative symptoms of schizophrenia, which you've probably covered in your other psychology classes. These positive symptoms include delusions, hallucinations, disorganized speech and behavior, but negative symptoms are things that are essentially missing from the person, like a flat affect, decreased speech, lack of motivation, and memory impairment. Schizophrenia is largely genetic. And there are also gene by environment interactions. So, for example, different environmental factors or triggers can potentially increase susceptibility to schizophrenia. But it is so much genetic that if your twin had schizophrenia, that means you would have a 50% chance of having it. The genes that we think are responsible for schizophrenia have to do with moderating synaptic transmission and synaptic plasticity. Still, the biological mechanisms are still being researched. Some hypotheses for this include myelin sheath defects or reduced cortical thickness. Interestingly, if you look at this picture from your textbook, in different case studies, we can see that patients with schizophrenia actually have enlarged lateral ventricles. In patients with schizophrenia, there's an increased number of dopamine receptors called D2 receptors in the brain. D2 receptors are expressed at really high concentrations in the basal ganglia, especially in the striatum, and they're thought to be important for motor control and learning. So when these receptors receive too much dopamine signaling, it can lead to psychotic episodes. Thus, antipsychotic drugs have been utilized to block dopamine receptors and specifically these D2 receptors. These drugs that are utilized for schizophrenia are effective in reducing positive symptoms like hallucinations and delusions. Of course, schizophrenia is a very complicated affective disorder because it presents both positive and negative symptoms. So researchers have been looking for drugs that could potentially mitigate both of these different types of symptoms. This brought along the glutamate hypothesis. So scientists believe that glutamate could be involved in schizophrenia. We know that glutamate is an excitatory neurotransmitter. The NMDA receptor is the main glutamate receptor in the brain and is both ligand and voltage gated. Remember, it requires both glutamate and membrane depolarization in order to activate and work properly. Interestingly, drugs like PCP and ketamine inhibit NMDA receptors and slow down glutamate signaling. Impaired glutamate signaling is implicated in schizophrenia. Rodents with fewer NMDA receptors exhibit schizophrenia-like behaviors, including impaired cognitive function. So ultimately, there may be overactivation of D2 receptors and impaired glutamate signaling via fewer NMDA receptors. So as you can see, there are many different biological mechanisms underlying these positive and negative symptoms of schizophrenia. Antipsychotics that block these dopamine receptors, known as D2 receptors, can be therapeutic, but they can have side effects. Tardive dyskinesia is one of these side effects in which someone will experience involuntary movement of lips and jaw. Additionally, Parkinson-like effects can be induced by this blockage of dopamine receptors. If you remember when we've discussed Parkinson's disease in class, Parkinson's patients have a huge loss of dopaminergic neurons in the substantia nigra. We know that dopamine plays an important role in motor movement and planned motor coordination. So sometimes blocking these D2 receptors can lead to Parkinson-like symptoms like tremors and difficulty conducting voluntary motor movement. Thus, research is moving forward trying to explore how increasing activation of NMDA receptors could increase responsiveness and potentially mitigate these positive and negative symptoms of schizophrenia. So potentially a combination of these therapies could be more effective, and this is further being explored in neuroscience research.